Hi, I'm Dr. Brian Kaufman, a family doctor and a CLL patient myself, and the Chief Medical Officer, Executive Vice President of the CLL Society, reporting some of the news from ASH. And obviously, we're doing this virtually, and I'm very lucky to have Dr. Mato with me. Dr. Mato, you want to introduce yourself, please?
Hi, I'm Anthony Mato, Director of the CLL program at Memorial Sloan Kettering Cancer Center, and it's great to be here.
So, Dr. Mato, one of your areas of research are finding new ways to help people with CLL who maybe failed some of the great new treatments we've had. And when something seems to be really important at ASH, it not only gets to have a poster presentation, but you actually get to stand in front of, in this case, virtually in front of hundreds, sometimes thousands of hematologists and present your really fresh data and take their questions. And you were lucky enough to have several of your research abstracts chosen to be oral presentations. But one that I wanted to ask you about is a new molecule called LOXO-305. So tell us why you think this is important, why it got chosen to be an oral presentation, and what a patient should know about this.
Well, this is a new class of agent called a non-covalent BTK inhibitor. And probably most patients have heard of BTK inhibitors before. Ibrutinib was the first, and acalabrutinib was the second to be approved for treating patients with CLL. But those molecules bind differently to the target BTK than this molecule, the LOXO-305, does. And what we know from the past 5 to 7 years is that molecules like ibrutinib, while they've changed practice and are very important, have their own limitations. A fair number of people will come off because of intolerance, or patients will ultimately progress over time because they develop resistance to the drug. And so initially, LOXO 305 and this class in general were developed to overcome the resistance to ibrutinib. It's a very, very specific target. It's a very targeted agent towards BTK, and it, because of the way it's designed and the way that it binds, it's different. And so theoretically should be highly active, should be well tolerated, um, should be dosed in a way that patients should be able to stay on the molecule for a long period of time and do well. And so this study, which is called the Bruin study, was a first in human, meaning these are the first people who ever took this molecule dose finding study where patients with CLL or lymphomas were treated with this molecule at doses between 25 milligrams and 300 milligrams per day. And myself and Sloan Kettering have been involved with this from the beginning of the study. And, just as a personal story, right from the get-go, I could tell there was something different here because I had patients at the lowest dose, 25 milligrams, responding within a few days, and they had seen many, many prior therapies. But the ASH meeting, I'll be presenting the data on CLL. Michael Wang from MD Anderson will be presenting it for mantle cell lymphoma. And in total, we're presenting data on more than 300 patients who've enrolled on this study over the last year and a half. 170 of them have CLL. And the results have been quite spectacular. And I'll just give you a couple of examples of what this population of patients look like. So for example, 80% of them had prior ibrutinib or acalabrutinib. So heavily pretreated, 3 to 4 prior therapies before they enrolled on this study, and lots of high risk features, including this BTK CYS481 mutation, which is the mutation that identifies resistance to ibrutinib, but other mutations as well, and other chromosomal abnormalities as well, like deletion 17p and p53 mutation. And so the summary is lots of patients heavily pretreated, and many of them had failed ibrutinib, venetoclax, and other agents. And so there isn't really a standard of care for that particular patient population. So like I said, we escalated the doses and were able to find a dose of 200 milligrams per day being safe for patients. So that's the dose that's moving forward. But
Is
just a
that
couple,
once a day, Dr. Mato?
yeah, it's once daily. And just a couple of comments about the safety. The drug was very well tolerated. There were, if you look at all of the side effects, we usually use 10% as a cutoff. There are actually only 3 side effects that made it to 10% or more, all grades, meaning even the most minor side effect or intensity of side effect, fatigue, mild diarrhea, and some bruising. Otherwise, all other side effects were less than 10%, and there still weren't very many. And the typical side effects that we care about the most with BTK inhibitors, like AFib, for example, occurred in less than 1% of patients in total. Only
Atrial
two,
fibrillation is the irregular heartbeats that we've seen with
correct.
ibrutinib and with acalabrutinib, maybe to a lesser extent.
Only 2 out of the 323 patients had AFib on the study. And significant bleeding was noted to be less than 1% of patients. And some other side effects like arthralgia, which seems to be, joint tenderness, which seems to be associated with drugs like ibrutinib, was only seen in 5% of patients. So it did appear to have a very well tolerated side effect profile. But even more importantly, the response rate for all CLL patients who were evaluable was 63%. But what I should mention is that takes into account all patients who've even just had one response assessment. If you start to look at patients who've been followed like 6, 8, or 10 months on the study, then the response rate goes up to 86%. So this is the first time we have data for a BTK inhibitor following another BTK inhibitor. And actually, we looked at the patients on the study, and we actually had patients who failed every other prior type of therapy. So it's like a small cohort of patients who had chemo, CD20 antibodies, BTK inhibitor, venetoclax, and a PI3K inhibitor. And still the response rate was not affected. And so, when you start to have patients that heavily pretreated with modern therapies and you're seeing responders, it's quite impressive. And to date, as part of the presentation, 94% of all the responding patients are still on this drug. And I have to say that a fair number of them were treated at our center. So for me, it's not only just a number, it's actually my patients. So, quarter or a third of these patients are my patients. And I can tell you, I'm in clinic every week, and they are very positive about how well tolerated the drug is, but also how it's working in situations where they didn't think they had a great option. So overall, it's early data. It's first in humans, certainly not FDA approved, not ready for FDA approval, but very encouraging data in a relatively large population of CLL patients who were treated with lots of other prior therapies, and this drug seemed active. So, in terms of my impression, I'm very enthusiastic about it and hoping for more confirmatory studies to help support the use of this class of molecules in general for helping patients with CLL.
So am I right in understanding that this binds differently than ibrutinib and acalabrutinib and zanubrutinib, which is on its way, but it works just as well for the people who have developed resistance to those drugs as to the people who are still sensitive to those drugs? Is there a
Correct.
difference?
There was, whether you were resistant or intolerant or never exposed to a BTK inhibitor, there were a few of those on the study. There didn't seem to be a difference in the response rate. And you're right, while the target is the same, it's probably not binding in exactly the same spot. And molecules like ibrutinib and acalabrutinib are irreversible inhibitors of BTK, meaning once they bind, they're stuck there. They don't come off. This drug comes on and off. It's a non-covalent binding drug, which means that the pressure for developing resistance is somewhat different with this class of molecules. And so that's why it's able to overcome the mechanism of resistance.
So two other questions, and you may not have the data on this. A rare reason that people become resistant to BTK inhibitors is a downstream mutation, PCL gamma 2. Was there any data? Are there any data on that?
There's only a few patients in total that had that mutation on the trial, only 4. There were, I don't remember off the top of my mind. I don't think any of them were independent, meaning that they didn't also have a BTK inhibitor mutation, but there were some responders with that molecule as well, with that mutation as well, which tells me that we probably have less of an understanding of what true BTK inhibitor resistance is than just having like a simple point mutation, and that explains it. It's a much more complicated story with subclones and alternative pathways that are upregulated and even different in CLL than other lymphomas. And so while I'd love to be able to say it's a simple story, you develop this point mutation and you're resistant, I think it's a complex story that we don't have yet the correlative science to explain why we're seeing what we see, but yet we are, I think most importantly, the patients are telling the story and that they're responding really well to this molecule.
I asked because I'm one of those rare patients who had a PCL gamma 2 mutation without the C481 mutation. So it does occur, but it's pretty darn rare. Yeah.
Pretty rare.
Yeah. What about Richter's? Well, any action in Richter's? Was Richter's an escape mechanism that you saw in some of these patients?
So there are definitely patients who were participated in the trial with Richter's, and there were more responders than non-responders. They're not
Wow.
they're not presented as part of the presentation I'm giving, and I don't recall whether or not they're part of Michael Wang's presentation, but we are in the process of developing the manuscript that includes all of the patients who participated in this trial, and there we'll be able to describe the response rate. I just came from clinic today and saw a patient who is on this molecule for Richter's, and he was already at 9 cycles of therapy. So really exhibiting a durable remission.
Any final thoughts or anything you want to share about this trial that patients should know? Is it ongoing? Is it open now?
It is ongoing in phase 2, and we're always happy to help either see patients and enroll them, or help them find a center that's closer to home where they could have access to this molecule if they're in a situation where it's appropriate.
All right. Well, we'll post a link to it through clinicaltrials.gov, and that should list where all the sites are. Dr. Mato, thank you so much. The things just keep moving forward and forward in CLL, and I'm so appreciative of the research you're doing. Thanks very much.
Thanks for having me.